So in this video, we are going to be going through our transport worksheet. So going through our matching the description, we have solute particles moving across the cell membrane from areas of high concentration to areas of low concentration. That is going to be diffusion. So going from high to low, just moving across the membrane, straight old diffusion. Solvent or water moving from areas of high to low concentration or low to high solute concentration, that is osmosis. Remember, there are two different ways we can think of osmosis. Water is going to be moving from high to low water concentration or low to high solute concentration. Next, we have movement of particles from areas of low to high concentration requiring energy. That is going to be our active transport. So active transport, the two things that we are going to be looking for to figure out if like an illustration, is active transport, we're going to be looking for it to be going from low to high concentration, and also that molecule of ATP signaling that we are using energy. And number 3, taking in particles through the cell membrane using vesicles. That is going to be endocytosis. So remember, endocytosis is taking stuff in through vesicles. Number 5, solute particles moving through the protein channel in cell membranes from high to low. That is going to be facilitated diffusion. So we can tell it's facilitated diffusion because it does not use energy and it is moving from high to low concentration. So it's following that concentration gradient. Number 6, removing particles through cell using a vesicle. That is going to be exocytosis. So we are using a vesicle to move stuff out of the cell. And finally, 7, solute particles moving from areas of high to low concentration without the use of energy. That is just simply going to be passive transport. So that is describing both our diffusion, osmosis, facilitated diffusion. Those are all forms of passive transport. Next, if we look on this picture, I wanted you to label the phospholipids, intracellular water, extracellular water, and protein channels. So we have our phospholipids here making our phospholipid bilayer. Our intracellular water is going to be inside, so inter means inside. Extracellular water is going to be on the outside of the cell. And then our protein channel is going to be right here. I realized I forgot a D. There we go, phospholipids. Hydrophobic, what does it mean? It means they don't like water. So hydrophobic is water fearing. The part of the cell membrane that is hydrophobic is going to be our tails, right? So these are going to be our hydrophobic fatty acid tails, or the inside of the cell membrane. Hydrophilic means it likes water, so hydrophilic means liking water or water loving. The part that is hydrophilic is going to be the outside or the phosphate heads of our phospholipids. A semi-permeable membrane. Semi-permeable means that it'll only let some things through, right? So it's going to be semi-permeable based on the size of the molecule, and it will, the size and like if it's hydrophobic, hydrophilic, stuff like that, it'll only let some things through. Looking at my picture of diffusion, we want to make sure that we are going from areas of high concentration to areas of low concentration. So I have a bunch of triangles over here on the left and only 3 triangles on the right. So we know that our triangles are going to move from the left to the right. Over here, osmosis, our squares are representing our water molecules. So our water molecules are going to move from areas of high concentration of water to low concentration of water, or you can think of it as low concentration of solute to high concentration of solute, right? So we are going to be our water is going to be moving from left to right. And then to figure out which one is hypotonic and which one is hypertonic, you want to look at the concentration of the solute. So whichever one has more solute in it is going to be hypertonic. Another way you can remember it is that water is just always going to move from hypotonic to hypertonic. So those are 2 different ways to look at it. On the back, we had 2 different Venn diagrams. Endocytosis is moving into the cell, exocytosis is out of the cell. The similarities of them is going to be vesicles and movement across a membrane. Oh, sorry, I skipped a question. On your osmosis from the previous page, what would you need to do to make them isotonic to each other? So isotonic means that they are equal. So what I would do is I would either add, I would need to make the number of water molecules and solute equal on each side, right? So I would add more solute to the left side and add more water molecules to the right side. Back to the Venn diagrams. So endocytosis moves into the cell, exocytosis moves out of the cell. Their similarities, they use vesicles and their movement across a membrane. Active transport, active transport versus passive transport. We are using, for active transport, we use low to high concentration and energy is needed. Passive transport, high to low concentration and no energy is needed. One similarity that they have is they can both use proteins, right? And again, they are going to be movement across the membrane. So that is your transport worksheet sort of walkthrough thing. If you have any questions, always feel free to let me know and I'll be happy to help.